Hi, welcome to this tutorial of PyEcoLib, a Python-based library to study stochastic size dynamics of E. coli bacteria. My name is Cesar Nieto, and I am currently working in the system biology group at University of Los Andes. In this tutorial, I'm going to explain first the mother machine experiments. The mother machine is the main device used to track single cell and to explore the size dynamics of E. coli bacteria. Then we are going to get started using PyEcoLib, simulating size dynamics and estimating the division strategy of the size division of E. coli. Finally, we're going to explain some additional properties of PyEcoLib, just like add different sources of stochasticity in our division and explore some different division strategies different from other. Finally, explaining some examples about simulating gene expression in E. coli. First, let me show you the people behind the project of PyEcoLib. Camilo Blanco, our engineer, me, Cesar Nieto, Juan Pedraza and Cesar Vargas, who were the leaders of the project. Okay, first, let me explain to you about the mother machine experiments, which consist in this kind of devices when you can put some bacteria in these trenches, and the bacteria in the bottom of the trenches are called mothers. These mother cells start to grow and divide, and the offspring start to running over the trench, and when they reach the end of the trench, they are filled away by the growth medium, which is liquid. Here you can see an example of actual microscopic photo of bacteria growing in these trenches. Size dynamics of the mother machine looks like this video, where you can see how bacteria grow and divide in some kind of stochasticity, and you can track the cell size along the time, discarding all the other descendant cells. So what you see experimentally is this kind of photos that you take along the time, where bacteria start to grow and divide. And the data looks like this, some sort of points revealing the growth of the bacteria and the division suddenly. The division times in bacteria data seems to be stochastic in some way. So you can merge some cycles like this, and the question here is how, for instance, the average size along the time for different mothers looks like, and how is the distribution, and specifically the variation of the coefficient of variation of the distribution. This is one of the main tasks that we do using a PyEcoLib. First, let me get started. What you need is installed the Python version 3.8.1 or higher. I recommend you to use the Anaconda prompt. These libraries, NumPy, CP, CPy, Math, and Platform are recommended to be installed before to run the PyEcoLib. You can only use the command pip install PyEcoLib to get the library. Let me show you how to do it. You can open only the Anaconda prompt, and using the command pip install PyEcoLib, you should get the library in this way. Okay, the current version is 2.0.2. We're planning to improve the library, so the version of this library will be started in next video. So you can learn about the library looking at our repository. You can Google it, searching for PyEcoLib GitHub, and the first result is our repository in GitHub. You can download the main code, and after you download it, you can use your Jupyter notebook to explore some examples. Let me see here the folder examples, and we can get started using PyEcoLib. The first thing you're gonna learn is how to use the simulator. For import the PyEcoLib library, use the command from PyEcoLib.simulator with P, E, and L capital letters, and simulator with no capital letters. Import simulator with as capital letter. This function, the simulator, is a class that needs these parameters. The first one is n cells. That is the number of cells you want to simulate. I recommend number of cells is 5,000, but it can simulate from 0 to a higher number if you have enough RAM memory. Year, which comes from growth rate, that tells you how fast do the cells grow. SB, which is the mean neuron cell size, that gives you approximately the scale of the bacteria during the simulation. Steps are the division steps that trigger the cell splitting. I'm going to explain you better this parameter in following slides. Other parameters, CV2 teeth, CV2 GR, lamp, and V0 array, I want to explain you later. The division steps here is one of the parameters that you need when you declare the simulator. The higher the division steps, in this case, for instance, 10, the more deterministic is your division. The minimum number of division steps is 1. If you use that number, you can see that the trajectories of the cell growth looks like if division happens more random than using, for instance, m equal to 10, where division has less stochasticity. Actually, the division using m equal to 1 have times distributed exponentially, and with m going to infinite, you have a perfect deterministic division. The first function we can use is the function SCT that comes from size dynamics. What this function does is to simulate many cells, the number of cells that you define in the simulator, and to generate stochastic trajectories for each cell, just like this. Then you need a Tmax that comes from a maximum time that tells to the program to simulate the trajectories from time 0 to that time. In this case, for instance, for this data frame, I simulate the size dynamics up to 5.4 minutes, for instance. The sample time tells you how often you take data. In this case, I take data every 1.8 minutes. This sample time takes the data periodically. So you take data every 1.8 minutes. And finally, to provide the name of the file you're gonna save your data. In this case, I use name CRM that comes from a continuous rate model. That is basically the main theoretical tool that we use to simulate these trajectories in PyEcoLib. You can find some examples in the folder examples, and let me show you how to implement this example. In the folder size statistics, you can find size statistic.ipymb, where you can find the libraries you're going to use. In this case, we use sys and os to make folders and to run into the directories in your main folder. NumPy, pandas for data analysis, matplotlib for plot graphs, and biaset that we're going to use to make some statistical inferring. Time, the chronometer that measures the time that the script needs to run. And finally, we import the PyEcoLib. PyEcoLib.simulator import simulator. And these are the meaning of the meaning, and these are the basic parameters of cell size. The mean size is going to be 3 micron. Doubling time is 18 minutes. Tmax is 180. Sample time is more or less 2 minutes. 10 division steps and 5,000 saws. Error rate is logarithm of 2 over this doubling time. Here we make these folders, one folder for the data and the other folder for figures. And first we define the simulator using the number of cells, the error rate, the mean size of birth, and the division steps. So we can run this SDIM using a sample time on 0.1 times the doubling time and exporting the data to this folder. If you see the data, you can do it using pandas and the function read CSV, and you can read the file, and you see how this looks like. The time in the first column and the sizes of every cell along the time. Here we use a statistical script to estimate the mean and the coefficient of variation of the size along the time using functions of NumPy, the variance over the mean squared, and we use this function of biases to estimate the confidence interval of those statistical moments, the mean and the C-square. In some cases, we want to estimate not a particular stochastic trajectories of size, but instead we want to estimate the trend line. I mean, how the mean size change along the time and how the variance change along the time. In this case, you can see that if we only consider stochasticity and diffusion times, we find that these oscillations in the mean size. So we implement this function SCD FSP with parameters Tmax, which is the maximum time, the CV square of the size if you start this dynamics from a given distribution of a starting size with this CV squared. By default, this value is 0. And finally, the name FSP is going to be the file where you save the data. The data looks like this. The time in one column, the mean size, and the variance of the size. You can compute the CV square of the size using variance over the mean squared. You can do it only run these lines, and is a little slower than the time spent to estimate the stochastic dynamics. And you can compare them using the cell, and you can see here some trajectories in ray behind the trend line using the stochastic simulation algorithm in blue, and the numeric using the function SCD FSP. The FSP comes from finite state projection, which is the numerical method that we use to calculate this trend line. And when you compare the results of the simulation and the numeric iterator, you see that both the size and the CV square are very similar, and actually they match perfectly. And you can find those oscillations that we can explain in following papers, how you can make it damped or more pronounced. Other properties that PyEcoLib can do and that you can find in experiments is division strategy. Division strategy tells you how bacteria decide to divide. The main paradigm that the scientific community have is that bacteria divides using the other division strategy. This division comes from when you compare the added size as function of the size at birth. In the other strategy, the added size doesn't depend on the size at birth. So if you start with a large cell, in this case the green one, the added size lambda, which is the size at division minus the size at birth, is the same than if you get born small. So what you see actually is this kind of cloud over the trend line given the stochastic fluctuations on the diffusion strategy. PyEcoLib can do that using the function diffstrat that comes from diffusion strategy. In the same way as a dim, you must define the Tmax, the maximum time to simulate the sample time, and the file where you're going to save your data. The argument is called name DSM, that comes from discrete stochastic map. that comes from the basic of this way to analyze the data, that in this case the map that takes the size of bird to the added size, which is a flat line. So this file is going to give you something like this: one column of the size of birds in that cycle, the size of division, the growth rate that can be stochastic, but I'm going to spare you how to do it in later, the cycle time, I mean the time between born and division, between birth and division, and the time where the division occurs. And if you can also obtain numerically the trends of the division strategy, what you actually find using simulation is this cloud. But using the function sdstat with argument of the size of birth, you can introduce the size of birth, and this function is going to give you the added size to plot this line, for instance, and the CV square of the added size. That's going to give you a quantification of the stochastic fluctuations over the trend line. And you can compare the simulation with this trend using this example, and first running the div_strat, and then using the function sdstat that comes from size at division statistics, giving an array of the size at birth. And when you compare using this script, you can obtain this this graph. Okay. If you're gonna go deeper to other capabilities of the PyEcoLib library, you're gonna go to other examples. For instance, if you want to add noise in division, for instance, the partitioning noise, which tells you that bacteria is not divided perfectly on a half, but on a random variable with mean 0.5 and some CV square, some distribution with a given CV squared, mean a given variance. And you can add also variability or stochastic variability in the growth rate of every cell. Since what you see experimentally is that some bacteria grow faster than the trend line and other bacteria grow slower than this trend line. How to do it? Here you can just add this additional parameters to the function simulator, to the object simulator. Two of the most important is v0 array, when you can put some a given size at birth, an array of sizes at birth. So if you want to simulate, for instance, 1,000 of cells, you have to provide a v0 array with 1,000 of elements. If the other parameter is CV2diff, which is the CV squared, the coefficient of variation squared of the septal position, I mean, the variance of this distribution over 0.52 square. By default, this division square, this CV square is 0, assuming that bacteria is perfectly half, I mean, split it on 0.5, the size of the mother. A typical value is 0.01, and that is low, but it's important if you want to simulate realistic division in E. coli. CV2GR that comes from the CV square of the growth rate is the parameter of the distribution of growth rates. A typical value is 0.02, and can be also important in order to simulate a realistic division in bacteria. So if you add those noises, if you started with this robust oscillation in this mean size that you get using considering only stochasticity in division times, if you assume that bacteria also have some variability in growth rate, using this case, and some variability in the position of the central ring, not perfectly on the half, this oscillation disappear, and what you have is this damped oscillations that we're going to show in oncoming paper, that this is what really actually happened in actual bacteria. You can implement this function in using the example noisy div. You can open it with the same parameters of size. You can add, for instance, this division CV squared, this growth rate CV square, and this size at birth CV squares. So you can produce this array of starting sizes with as random numbers with distribution in gamma, and with this shape parameter and this scale parameter that comes from this CV square, and with the number of cells you are simulating, in this case 5,000. And since these numbers have mean 1, you can just multiply this array times the mean size, and finally you have the mean size as you want, for instance, in this case 3 micron. You now declare the simulator using these parameters, CV2 div, CV2 growth rate, and the V0 rate you defined here. And you can use the function sdim to produce some stochastic size trajectories along the time. You can, using this script, you can estimate the statistic mean and the CV square of the size along the time, and what you get is this trend of the mean size along the time, which are damped oscillations, and the oscillations of the CV square of the size along the time that are also damped. Since we put this variability in growth rate and division parameters. Other things that PyEcoLib can do is consider division strategy different from other. So the other are the most important division strategy found in PyEcoLib. You can find other division strategies that can found in some circumstances like when bacteria grow in minimal medium or in a poor nutrient growth medium. they seems to be some kind of sizer. So let me explain what are these division strategies. The other, as I explained before, tells you that bacteria have an added size before division that doesn't depend on the size at birth. On the other hand, the timer strategy tells you that bacteria wait a given time to divide. So if you get born large, you're going to divide larger, and if you get born small, you divide smaller, not smaller than considering, for instance, the other strategy. So the relationship between the added size delta and the size at birth is actually a positive slope straight line with some fluctuations, stochastic fluctuations. In this case is a flat line, and in this case it's a positive slope straight line. If a third division strategy is the sizer strategy, that tells you that bacteria want to reach a given size before division. So if the bacteria get born large, they add a small quantity of size because they are closer to that objective size. If the bacteria get born small, they have to grow more in this case, so the added size is larger than the case where bacteria get born big. So you can see here that what you get in the sizer strategy is a negative slope plus some stochastic fluctuations. The larger you get born, the less you add before division. We demonstrate that you can obtain any of this distribution of this division strategy changing one parameter lambda that goes from timer when lambda goes to 0. The other is using lambda equal to 1, and the perfect sizer is using lambda equal or going to infinite. A PyEcoLib can reproduce these division strategies using lambda, but for numerical stability, we recommend you to use lambda an interval from lambda to 2 to lambda to 0.5. In these cases, the division strategies are timer-like for lambda from 0 to 1, and sizer-like with lambda from 1 to infinite. What you have to do is to include in, when you declare the simulator, the argument lamp equal to 2, for instance, to obtain the sizer-like strategy. By default, this lambda is 1, that gives you the other. So if you don't use this argument, the program is gonna give you the other division strategy. You can use the same functions, for instance, the divstrat, to actually see if the slope of added size as a function of the size of birth is negative, and the function as the stat to obtain the numerical trends of the division strategy. And finally, you can see something like this. The added size of function of the size of birth is gonna look something like this cloud, and with a negative slope relationship. And the CV squared is gonna be a positive slope as a function of the size of birth. You can find this example in the folder examples and sizer-like. You can open the notebook sizer-like, and using lambda equal to 2 and the same functions, you can obtain also the SDIM FSP, the dynamics using numerical methods of the mean size and its variability. You can obtain here the division strategy, and also you can compare the mean size along the time and its variability along the time, and you see that numerical method and simulations match perfectly. You can use different lambda, and you can compare them, the trends, using other more advanced examples that you can find in the folder Advanced Size Statistics. When we present how to compute the dynamics, for instance, here, obtaining different noises. In this case, for instance, you can see how looks like this stochastic division, I mean, with no additional noises, how looks the size and how looks the CV square of the size along the time. What did you consider that bacteria, so growing at the same growth rate and dividing exactly on a half, what did you consider finite initial distribution? I mean, not starting all of them at size 1, but from a given distribution with a mean equal to 1. You see that the oscillation are have less amplitude, but they are robust. On the other hand, you consider noise in splitting. I mean, but bacteria do not divide perfectly in a half, and they grow with a given distribution of growth rates, how the oscillation are damped. And what did you consider different division strategies using lambda equal to 0.5, which is timer-like, lambda equal to 1, and lambda equal to 2, which is the sizer-like division strategy? Okay, you can compare all of them, and you can see, for instance, in the gray, in the background, some of the stochastic trajectories that you can compute using this PyEcoLib library. Okay, and you can also study the autocorrelation function of these trajectories that we can explain in following papers. Finally, we present an example about how to simulate gene expression using PyEcoLib. You can find the folder in examples as well, and the folder is called Gene Expression Direct SSA. SSA comes from a stochastic simulation algorithm. You can open the notebook. First, you define the size parameters, the growth rate, and these are the parameters of the gene expression. In this case, we simulate a housekeeping protein, a constitutive protein with a messenger RNA intermediate. We define the parameters such as the cell have 10 RNA molecules as a mean concentration per femtoliter of per micrometer, if you prefer to use the length instead of the volume. Protein concentration with average 100 of proteins over at a given unit of volume. The degradation rate of RNA, which is 5 times the growth rate, I mean 5 times faster than the growth rate, and the production rate, the transcription rate, that depends on the steady state concentration of RNA and the dilution rate, which is the growth rate, and the degradation rate. The translation rate that gives you the rate that proteins are produced. And we explain deeper the algorithm that we use to simulate this gene expression in our supplementary material. And I'm going to explain you very briefly what this script does. First, I define an array of population with a given number of cells and 6 properties. One of them is the size. The other one is the number of the RNAs, the number of proteins, the times that of the chemical reactions that you can compute it using Gillespie algorithm, and the last one is going to be you can use the function get next t that gives you the next time that and the next division step is going to happen. Okay, so what we do here is to make this iterator over the entire population, and I define a simulator of one cell with the given parameters of the size. We calculate these parameters, and we have the reaction matrix here. We have number of RNAs, the number of proteins from time to 0 to T max, the size, then time to the next division step, and we put all this the times of the reactions, the RNA creation, the RNA degradation, and the protein creation. You can see here that you don't have the protein dilution, since if you consider the division using PyEcoLib, every time the cell divides, the number of proteins is gonna halve. So you don't have to worry about the dilution rate of the proteins, neither the dilution rate of the RNA, but just only worry about the degradation rate, the active degradation rate. So you take in those times, you get the minimum time of these 4 times, and taking that minimum is going to give you what reaction is going to happen. Okay, so we simulate our size dynamics until that time, and if this minimum time is about some chemical reaction, so you perform the reaction, in this case adding 1 RNA, destroy 1 RNA, or produce 1 protein. If the minimum is at time of division step, you can see how much division steps the cell 0 in this case have performed at that time, And if the number of division steps is less than the division step that is higher than the division step you have on objective, you divide and you segregate your RNA using a minimal random variable in both the RNA and the protein using this dp, which is the division parameter. I mean, if the division is considered perfectly on a half, this division parameter is just 0.5. But if you consider that division is not perfectly deterministic on a half, but with the given CV square of the division, you can take this division parameter from the function getDP, and you can put it in the as parameter in the binomial distribution. You take the size that was calculated using the function simulate that simulates the entire trajectories at a given time, during a given time, the new number of RNA, the new number of protein, the new sizes, and you can compute the new Gillespie chemical reaction times. And you can obtain, for instance, the concentration of RNA just by dividing the number of RNA and the size, the protein concentration, which is the number of proteins over the size, and the numbers, if you want to make some statistics, the number of proteins and the number of RNA and the number of proteins as the size as well. You can obtain here, for instance, the size dynamics, the dynamics of the RNA concentration. This is the dark red solid line, and some pink or pale red trajectories, and the CV square, how this looks like. You can see here that you can see some oscillation of these CV squares. You can also see here the number of RNAs along the time, and some trajectories of some cells in the background, and how the CV square of the number of RNAs change along the time. The protein concentration as well, and some trajectories of the protein concentration along the time, and the CV square of the protein concentration. The number of proteins as well. Since bacteria are growing, you can see that this variable is very correlated to the size. And you can see also the coefficient of variance of the number of proteins. And you can compare all of them. Okay, that's all. Thank you very much for your attention, and I hope you enjoyed this PyEcoLib library. So thank you very much. See you in the next tutorials.